All right. So I get number 1 this week. It's a little complicated, but we're going to try and simplify it. So this is kind of a visual representation of what it's kind of talking about. We're given the mRNA, which is this that's been coded. These other 2 strands are DNA. It's been split apart, and you get your template and your coding strands. So that's kind of how that's coming in, and then this is how, or what we're given. So our mRNA is here in the middle, like up here. And we're trying to find out our coding and our DNA templates. So our DNA template is going to be the same thing as our mRNA, except we're replacing our uracil with our thymine, or U with T. And your coding is going to actually go through the synthesis process and find the base pairs that match. So there's that. And then D. So in the last final question, it's asking about where kind of the diagram, as you can see, and then where transcription or title box is going to be. So we believe that it's going to start on the right side, so over here, because if it's going this way, then it's going to be on our end. So it'll be on the 5' end of our template.
All right, so I have the remainder of the questions, so bear with me for a little bit. So for 2, that was our vocab, fill in the blanks. I'm just going to give you the answers that we got and spare you the time of reading the actual questions. We've got for A, we got our Tata binding proteins and our sigma factors. B, I can't even pronounce it, I'm going to completely botch this, but we've got guanylyl transferase, oh my gosh. C was group 1 introns and group 2 introns. D was the CPSF. E was the TF2B and the TF2H. And then F, we got our SNRPs and our SNRNAs. For 3, it talks about indicating which 2 proteins in either prokaryotes or eukaryotes that act as helicase. We've got RNA polymerase and TF2H. B, it asks for 1 protein that acts as a helicase. We've got the RHO or P protein. For C, it said indicate 3 molecules in eukaryotes that function during RNA modification as whatever. We got group 1, group 2, and SNRNAs. For D, it said indicate 1 protein that acts as a transcription factor. We got our sigma 70, or our housekeeping sigma factor. For E, it asks for 1 enzyme in eukaryotes that synthesizes a long string of identical RNA nucleotides, but does not use a template DNA strand for RNA synthesis. We got our SNRNPs, and we just included the SNRNAs in there because they work together. They're each part of each other. For number 4, just based off of time and there only being 2 of us today, we decided to allow you the chance to not suffer through number 4 twice, since how our gracious instructor has already given us the way to work through number 4. So for number 5, for A, it asks which of the above is found in the bacterium E. coli. You've got the -35, the RUT, and the NUSA. For which one was found in the yeast, it asks, well, obviously you can subtract the rest of them, or subtract these from the rest, and we're going to just add the rest to the list. We've got our -25 enhancers, silencers, TF2D, the SNRPs, GC box, and our group 1 introns. For C, it asks which of the above is involved in the transcription initiation. We've got our -35 and our TF2Ds. D, it talks about which of the above is involved in the transcription termination. We've got our RUT, NUSA, and our SNRPs. For E, it asks for which of the above is a DNA sequence. We've got a -35, -25, and a GC box.